The meeting's being recorded. You just have to press continue. And this meeting will be, recording will be available to all of you on YouTube. We'll let you know where it is. I'm really excited that we have Nirav to speak to us today. Nirav is the director of the University of Arizona Data Science Institute, and he has a background in industrial and systems engineering. And really his interest is in computational platforms that can be scaled to support open science and innovation. And I have learned so much in my short conversations with Nirav, and my appreciation for data science has grown tremendously. So I am really looking forward to your talk, and take it away.
Thank you, Reina, and thank you, Rosha, and thank you, students, for giving us the chance. I'm here along with my colleague, Anthony. Together, we are part of what's called DMAC for the Superfund, and I'll say a few words about it, but I will get started. Let me just share my screen.
Can you share it?
Yeah, I am just about I have so many screens that sometimes I'm not sure which one I'm sharing. Yeah. Can you guys see it as a screen? Perfect. And I am going to move a few things around so it's easier for me. So what I wanted to talk to you about today is concepts for reproducible research and good enough practices in scientific computing. And this is going to give you sort of a quick flyover of what are the tools, what are the methods, what are the techniques. And all of these are available to you, accessible to you, especially on our campus. And so I want you to sort of make you aware of what's there and how, if you're not using some of these, you could go about using them. And at the bottom, you sort of see examples of things like SnakeMake and Docker, and I'll talk about all of those things in quick succession under the concept of what is reproducible research and what are good enough practices in scientific computing. So the content that I'm going to show you, a lot of it is handiwork of my student Satish Perry, who also works on my NSF Sweeter grant. Sweeter is the Southwest. We formed a consortium of different universities, and it's called Cyber Teams, all the way from Texas to New Mexico and Arizona and California. So UC Berkeley is part of our team as well. And what we do is try and promote best practices in scientific computing and data science. So Satish has been organizing lots of small workshops on campus on these topics. So this material also comes from his workshops. And a big shout out to the National Bioinformatics Infrastructure of Sweden, which also has put together excellent documentation in this topic. And I have the URL for their site as well. So please do visit and look at the work they're doing. So as I said, I'm part of DMAC, the SRP grant that we are very fortunate to have on our campus that underwrites a lot of these activities. Sangoi, who is teaching today at the same time, is our fearless leader, and that photograph of him is before he got tenure. So his professorship has taken a toll on him. I pretty much look the same. Michelle and Anthony are our engines that drive DMAC. Michelle's our software engineer, and Anthony is also, I would now start calling Anthony a software engineer because he's writing so much software and he's a statistician at the same time. And then DMAC as a core, what we focus on is how do we make data storage, processing, analysis more accessible to the members of our Superfund. And then we are building methods for predictive algorithms that are easier for people to access, and then analyze that data using those methods. And finally, we provide those as web applications, as I was telling you. And it is relatively easy for people to now analyze fairly complex data using these methods. So now let's talk about what is reproducible research and why people are talking a lot about it. Well, in 2015, there was a paper where for the psychological research, and they looked at reproducibility of experiments. They took 100 experiments, and they found about 1/2 to 2/3 of those could not be replicated at all. And so that was like a alarm bell for their community. And then soon after, every other community started waking up and saying, oh my God, a lot of the analysis we read in our papers are not reproducible by anybody other than the person, and possibly only on their laptop and nowhere else. And so this became a reproducibility crisis. And then you can see of the various communities where people had a hard time reproducing someone else's research, and often their own research. And this is not a, I'm not trying to make a joke out of it, but you will always have a hard time going back in time and redoing the same analysis you performed if you don't have the scientific rigor, the bookkeeping, the accountability that goes with it. If you're not organized, your prior activities are going to bite you in the future. So you always have to be kind to your future self by organizing your data and computation. So you're doing it, think of it as a selfish thing just for you. And if you can be happy, the community around you will be happy using what you do. So you can see in chemistry, in biology, in just about every discipline, you're looking at somewhere closer to like between 80 and 90% to 60 to 80%. Your own work, let alone somebody else's. And so when people are asked, is there a reproducibility crisis? And most scientists said yes, and I would encourage you to go take a look at these papers. And so this is a genuine crisis. It's not something that we're just talking of the sky is falling. It is a problem. And very often authors move on, and the lab that you were in is still working on things you've done. You may move on to doing other things, but the student that comes after you, somebody else that's building on top of your work, is now on shaky grounds because a lot of those things were not accounted for. So replication of data analysis in 18 articles in microarray, you can see right here what goes in and how hard it is. And so a lot of it on the pie chart, you can see they just can't reproduce it. And some in principle, some with some discrepancies, and some with partial discrepancies, some from process data and some discrepancies. But all in all, this is not a pretty picture. So the summary of efforts to replicate published analysis is hard, and it should not be that way. And here we are trying to talk of what people are doing with somebody else's data and their methods, let alone bring in new data and use the same method. Typically, when you read a paper, you're now going to take what they did and then use that to process your own data. And so a lot of this that cannot reproduce comes from software not available, method is unclear, the results you get are different, and a big chunk is data is not available. And you really need to solve this holistically to be successful. So why is this a crisis? What really is causing this crisis? So when you look at the current workflow, this is how people do analysis. Okay, so the author has a local machine. The machine could be a Mac. Then they used a version of R. They used a certain version of the genome. And then they found, let's say, in this case, 223 significant genes. Then the reviewer or reader A is using Ubuntu, which is a Linux flavor. Their version of R is a little bit different than what the author did. and they are using a different version of that genome. This is version 18, this is version 19. Now they're finding 228 significant genes. The next person comes along, they are using a little bit older version of the Mac OS, 10.10.4, but then they have a newer version of R, and they have an even newer version of the reference genome. They find 227 significant genes. We are doing exactly the same protocol, following the same programs, but there are minor variations in version 18, 19, 20 of this reference genome. And so these are the kind of things that cause a lot of angst in the community. So how do you sort of avoid that? There's a whole technology called containers, and I'll talk a bit about it, but think of it as packaging everything you have in a lunch box, if you may, where your lunch bag, where you have your food, your salad, your utensils, your paper napkins to clean your face, your dessert, and you put that in your lunch bag. Now you can go take that lunch, eat it on your desk. You take that lunch, eat it outside in the garden. It should pretty much be the same lunch. It should not change because you moved your location. And so containers are a way to organize your operating system, your programs, the version of everything, and then give somebody that container rather than say, here's my recipe, figure it out. And you can think of the recipe as if somebody gave you a recipe to cook something, there's enough variation that you have while cooking that when you follow, there are instructions that are not exactly what the author anticipated. You have your own version. And I love to cook. And when I cook, I hate measurements. There are some things I measure, but the rest is how I feel. And I'm looking at something and I'm like, I'm going to add more spice. It feels that this would do better with what I'm cooking. And those are the assumptions that come into scientific computing as well. That's just human nature. And so when you make these containers, the recipe is locked in, and then you can give the container to anybody else, and they are running the same container. So you're going to get the same answer. So containers are a very big part of your life. And those of you who don't know what those are, you should get to know them, and we will have some training for you for that. So then what are the tools for reproducibility? So at the base level, we talked of Docker as in containerization as a technology. If you're running some of these on UA HPC, you have something called Singularity. If you're using R, then there is something called R Markdown. And then there are some things called Jupyter Notebooks. These are all interactive ways of writing your steps one by one along with instructions. When I give you my notebook, just like a physical notebook, and if I've kept detailed logs, you can then do what I have written in the notebook and reproduce my experiment, whether it's a real experiment at the lab, on the lab bench, or my analysis. And so Jupyter Notebooks are geared towards that analysis or R Markdown, where your code and instructions coexist. So you're not reading a separate piece of paper and then typing. You pretty much see everything and you can click and say next, next, next. So if you don't know what notebooks are, you should get very friendly with them. The layer above that is something called workflow management systems. Often for beginners, we highly recommend using SnakeMake, and you can see a little snake here. And SnakeMake allows you to describe the steps, and a workflow manager is very smart in knowing where things break. And if they break, it's not going to redo the steps where you already have an output. It'll only pick up where it fell down and then do the work forward. And when you give somebody that workflow file, they can very quickly redo the steps. And then Conda is an environment where you can set up these virtual environments, much like containers, where everything that you need is put together in a ready-made way. And then on the top layer is something called Git. This is how you control the version, because as human beings, we keep doing iterative things. We start with the analysis. We want to improve it. We want to further refine it. we share it with someone, they give you new ideas, you improve it. Now you should be able to go in time, almost like a time machine, and say, I would like to do what I did last Tuesday with this piece of code. But if you're constantly overwriting it and just saying, my awesome program, then you don't have an awesome program from last 2 weeks. But if you're using version control, it keeps a copy and it knows what changed. And you can say, what the hell was I doing last week? It worked. This week, it's not working. Can I compare those two versions and think, oh, I made a change here and that's breaking things. I'm going to fix that. So version control is really essential for what you do. Given we are a smaller group, I'm happy to sort of pause anytime people have questions, or you can stop me and ask me questions. So please feel free. So here is sort of another paper on analysis of journal policy effectiveness for computational reproducibility. So if you, certain journals will ask the author to provide the data and the code. And so this group, Victoria Stoughton and her team, went ahead and looked at what is the impact of journals doing that. And they contacted many authors, about 180 responded. And you can sort of read their analysis here. A lot of people shared their data in code. Quite a few of them didn't respond. Some of them didn't share the data or code. And some just said, no, I'm not going to share that with you. And many of them said, go read the supplement that we have included. So you can see 36% were willing to share their, that's not very encouraging, but that still is pretty decent. But you can see some of the things where it was, I used my code and there's no public version. Since this code is not very user-friendly and is under constant development, I prefer not to share this code. This is no longer an option. You really have to be able to share your code if you want to get published in good journals and go through the review process and survive. And then here is, we do not typically share our internal data or code with people outside our collaboration. And when you approach a PI for the source code and raw data, you better explain who you are, who you work for, why do you need it, and what are you going to do with it. And I have to say, this is a very unusual request. And without any explanation, please ask your supervisor to send me an email with a detailed, and I mean detailed explanation. This is not collaborative. This is the reason you need open science, you need open data, and then you are able to get these things and extend somebody else's work and feel confident that if you're basing your science on somebody else's observation, you give them the absolute credit, but you should be able to verify and then validate and then move forward. It should not be, please take my word, I'm awesome, this research is awesome, just use it. And so, what is available for inspection, you can see from 2009 to 2010, 52%, but by 11 and 12, a lot of these things are getting more and more available. All the major software and the locations given, the data locations are given, so people are changing. And so, as I said, if you want to be, and this is from 2012, so by now these numbers have really shot up. So if you want to be successful, you have to follow these practices. So what do you mean when something is reproducible? So if you take code and data, if these things line up, then you get reproducible. So if it's the same code, but a different data set, you call it replicable. I can replicate this research. If the code is different and I take the data from the same person, it's considered robust. If the code is different, I take that data, it's called generalizable. But here in reproducible, we are saying if I take the same data and the same software, I should be able to get the same answer. And so the environment where you do this is really, really critical, where the data comes in, the code comes in, and that environment is exact, and hence the results are exact. So this is sort of where we are focusing right now. So the foundation of knowledge should be constituted by experimental produce facts which can be made believable to a scientific community by their reproducibility. And this quote is from Boyle from the 1600s. And here is the NBIS code, which I really like. It's like, why are we calling this course reproducible research? It should just be called research, because if you're doing it right, it should be reproducible. So now I'll say a bit about FAIR, which is the findable, accessible, interoperable, and reusable. That's what people are striving with what they do. And so you have to look at where is your latest publication fitting in. And I'm not going to go through each of these, but you can see that if the data is available on request, all the code is available on request, and the instructions are there, it's considered decent. But if the data is in a public repository, all the code for it is available, and it's available in a timestamp format, and all the programs used with their respective versions are available, you're getting there. If your paper has a section to aid the reproduction, and you say, this is how you would do it, you have used non-proprietary methods. You've used CSV instead of XLS. You've used formats that are available as open, and that's better. And all the code is available in raw format, it's documented. And even if you're using seeds for something, for random number generation, all of that is saved. And the instructions for reproducing the environment are publicly available. This is where I want all of you to be. This is where we want to take you as a community. And this is sort of the good enough practice for scientific computing that I'm talking to you about. And so here is example of something from recent work. And so often people ask, what is it? Why should I do it? How am I going to benefit? And so, as I said, if you didn't take some of these training, when you look at your reviewer comments, when they come back from a paper and they ask you to make a change, you're like, how did I do this analysis? How did I get here? And you start thinking. But if you have followed these protocols and processes, and the reviewer says change something, all you have to do is modify some parameters and hit run or reperform that analysis, because it's a whole lot easier. And so, the improved structure, the organization is really important for you. And so you want it such that your manuscript almost writes itself when it comes to the analysis part. And then you can pretty much reproduce it, but also resume it. So if you've stopped it, done something, and you come back, it doesn't take you that long to figure out what the hell was I doing. And once you start making things more reproducible, people start trusting you, and you're going to become a lot more wanted colleague that people want to include you in activities, because your quality of work, the finish of what you do, is at a very different level. Okay. So now I'm going to switch gears on you and talk a little bit about how do you manage that data. So data mismanagement in practice. So often we get raw data, it arrives in some cumbersome proprietary format. So that's data acquisition. The metadata, which is data about data, sits in the researcher's lab journal. So you may say, I got my data from sequencing today, and you keep that in a journal. Then you do the analysis, and it gets converted into the format of choice. The original file, the conversion settings are lost. So what you got in this, by the time it comes to analysis, is lost. And a lot of the hard-coded variables are in the scripts that you use, and they don't reach the metadata stage the way you want. Then you submit the first submission, and then you send things back and forth with your collaborator, but you include nice Excel sheets that are colored so you can keep track of things. Then when your paper is in review mode, it leads a quiet life on the HPC cluster or somewhere where it's being analyzed, and it has to be sort of urgently reviewed. And then when you do a second submission to that, it ends its day on an external hard drive sitting below some researcher's desk. And then that same metadata is reformatted and included as a PDF in the supplementary part of your paper. such that nobody else can reuse it. And then your paper comes out, and your best bet is to say data available on request. Now that is not where you want to be. This is old way of doing things. Today, I can assure you many journals will reject your paper if that's how it's managed. So where does FAIR come into play? So here is the link to the paper. I would encourage everybody to read that paper. And it actually has really nice guiding principles and how do you make your data findable. And it's not just about searching, but it is what do you add to it such that it is more findable. And the accessible part is what standards do you follow? How do you make sure that it's accessible? And the interoperable part is connecting it with things, staying consistent. Even simple things like naming your columns. If you're going to not follow the standard or set a culture for the lab that you are in, you and your colleague cannot exchange those spreadsheets or things that we often call as data frames in R or in Python, where you put those things together. So it's important to have all your colleagues, whether it's in your lab or your cohort that's working together or a community that's performing analysis. If you all don't agree to some of these things, it becomes really hard to share each other's data and perform analysis and be interoperable and be reusable. So everybody talks of writing a data management plan. We all know our funding agencies want it. There's a good reason why they want it. And you sort of go through this life cycle of scientific data analysis. The challenge is most people obsess over the last step, which is I am going to write this data into some sort of institutional repository that when I publish it, let's say for example Figshare, I'm going to get all excited and say I'm going to make this beautiful institutional data repository where I'll put the data, and once it gets there, it looks so nice, everybody will be happy, and I'm going to get fame and fortune. That's the last step. You should do it, you have to do it, but to reach that, there's a lot of ugly, dirty work. Like there is a lot that you have to do in between where your data is going to go through these different phases. And if you don't obsess on that step, you cannot make it to the last step of the institutional data repository where everything that you need looks presentable and you're proud of it. You're going to treat that as an institutional dump yard where you're going to run and say, I need to graduate. I just need to deposit my data. So take it and leave me alone. You can't have that happen. So imagining every step of your life cycle is critical and you have to plan for it. And so here are some steps, and we can always come and talk to you more about each of them. Finally, sort of data acquisition and depositing, I gave you some examples. Always think of what SRA or GEO or whatever is your dedicated or canonical institutional repository. You want to make sure that you are preparing for that. If I need to send it to the U of A data, which is like our institutional data repository, I need to make sure that I'm meeting their standard. If I'm producing short read archive systems sequences, I need to know what is the meta information I need, because when I'm ready to submit to them and I haven't planned it, it is going to be a problem. And you have everything from data Dryad, Figshare, Zenodo, as I mentioned, and try and get a digital object identifier so that you know where your data is and how to find it. So when you publish it, it's easy for others to get to it. And then structure the metadata in a format like your repository asks for. As I told you, if you don't plan for this upfront, you're going to hurt subsequently. And you can see SRA uses a template, Geo also has a soft format, and then stick to non-proprietary and widely used file formats. Do not reinvent the wheel and say, it's super easy if I do it this way, I'm going to be the pioneer and come up with a new format. Please don't. Honestly, please don't. And we have lots of formats. Find out if something is missing. And if it's missing, talk to the authorities that have it and put a request for improving it. If it doesn't get improved, then get a community, but don't become your own leader and create formats. Similarly, you can see lots of places do not share data. It's hard. Find an open access place that'll take your data and then make it available. And that's where you get the democracy and transparency. Other people can work with what you're doing. And then the citation goes up when your data is available. And as we all know, those are important metrics for everyone, the students, the faculty, the projects. If we don't have good metrics on our performance, it's very hard to quantify our impact. So subsequently, getting more funding becomes harder. And towards the end, I'm going to just say a few words about how do you structure your project? How do you organize your assets? So a lot of you who are interested in data science, I would highly recommend looking at the cookie cutter data science, where it teaches you how you can quickly just organize your data. And using those tools, it'll make all the subdirectories for you. So whether it's data, so if it's external, interim, processed, raw, all the documentation, where do you put your Jupyter notebooks? Where do you keep your references, all your reports, figures? So when you're writing your paper, you don't have to go hunting and pecking. And if I say, hey, come, please help me with my analysis, you land in a known environment where everything that you need is in well-known places. So please follow like a structure that makes sense. Again, start with your lab, start with your colleagues, start with the community you're part of. If they don't have a standard, help them come to a standard. And these standards are not very complicated. And so the good example here is, you have data, just don't call it sample v2 and create one big pile. You can start saying, I'm doing RNA-seq, this is post-sequencing, the adapters are here, the FastQC results are here, the HT sequence counts are here. So making sure that what you need as a community are all in some sort of organized manner. And the first step towards reproducibility is getting organized. And you divide your work into distinct projects. And every time you get a new project, you create that new cookie cutter template and include everything in one location. And you can see what I showed you with cookie cutter data science and this, it's very identical. But you can see here is SnakeMake, the workflow files, the Docker files. And this is the paper, again, that I was mentioning, a quick guide to organizing computational biology projects. But honestly, you can use it for different things. Here's the GitHub repo for that. And they have a template. And making sure your toolbox of what you use is consistent. For example, use a good editor like VS Code and then write things in it. Or if you're using RStudio or Sublime Text, make sure you organize your text files and code using Git. And then file naming conventions are critical. Don't use crazy characters that are special. Avoid spaces because a lot of programs like to have no spaces, especially Linux programs. Also know the content of the file without opening. So if you have certain things, try and put them in the file name, but sometimes that's too hard. And then control the file ordering, use dates when possible, and use 0, 1, 0, 2 rather than 1, 2, so that when you sort things, they're easy to find. And then here are some bad examples where you have such things in file names. These are the good examples of file names. And same things with source code. So make sure you do this kind of organization. You have your raw external data, internal data, and anybody should be able to reproduce. And so finally, going back to this, everything should be in this column. And this is the paper which talks of good enough practice in scientific computing. I recommend that to everyone. It's by Greg Wilson, who also started the software carpentry movement. And all of you should try and attend some of those workshops and get those skills. So with that, I will stop and take questions.
Thanks, Nirav. Let me start with a couple of questions that maybe aren't so much about your content, but about the availability. So can we make these tools that you've developed available to other Superfund centers?
Absolutely.
Okay.
And so, Reina, as when we were writing this, all the logins to access the tools that Anthony and Michelle and Xing are creating, all you need is a Cyverse login to get in to start using them.
Mhm.
And they are meant for not just the Superfunds, but it could be your collaborators in any country. So we are all about open science and open innovation.
And so how do we best communicate, do you think, to NIEHS about the availability of these tools?
Good question again. What we need to do is first, now these tools are ready, is to have small workshops, and we've already, Michelle and Anthony have geared up for it. And so we're now going to start the training aspect. And then as we get our own students, our own cores and teams in our SRP, starting to use it, give us feedback, and then this becomes part of their ecosystem, then we can go out to the larger community and say, this is how we are doing it. This is how it works for us. You're welcome to participate in, using these tools or building new tools using the same templates so that there's interoperability between our tools.
Mm-hmm. And so what do you think that time frame is for this sort of beta testing here?
I'm going to put Anthony on a spot for a little bit, and then I'll answer it, but it's more fun to harass Anthony.
Yeah.
Anthony, how is your tool looking for somebody else to use?
Well, I think we're expecting to do the first presentation and kind of intro to it here in the next month or so. So as far as I know, that I think that's kind of the timeline.
And is that the internal Anthony, or external?
Oh no, internal.
Internal.
And so what would you think might be an external timeline?
I think it really depends on how the internal goes.
Okay, yeah. Fair enough.
That's the best answer I can give you right now, sorry.
I only ask because it's my job to sort of make sure that this goes beyond U of A, because I think this is so exciting what you're doing, and you explain it. It seems so simple when you explain it, Nirav. But I think that it will take some time and effort to communicate to NIEHS that this is available, and to set up some webinars and things like that. So I'd like to know when I should start that effort.
Yeah, I wish Xing was here, okay. But he had some ideas of time frame. I would say this summer was sort of us getting our feet on the ground and being solid, because a lot of the technology that we are using, that Anthony and Michelle are using, once we have a hang of it, because when people will come in, they'll want to do their own thing also. And so we want both Anthony and Michelle to have enough training bandwidth to answer some of the questions. So if we don't take care of our internal projects first, it'll be hard if we get consumed in external projects. The other good thing is a lot of the training material to come up to speed for utilizing this exists in Cyverse, and Michelle and Anthony are getting more and more comfortable with that. So what will happen is we'll pull in some colleagues from Cyverse, take that material, and then do special workshops for our Superfund sites, let's say on containers and how to run the containers, notebooks and R Markdown, how to use that. And I would love for our own students from this course to be the template, or say, this is how I'm using this for my project, because hearing it from a peer group is way better. Like what you said, when I say it, it sounds simple and it's marketing. But when the real scientist does it, they talk a lot more about this is broken, that's broken, but this works, that works. And that is what most genuine people look for. So I'm hoping that as we get our internal team comfortable with it, we can then have a peer group that they work with. And so that way, this is made available to our broader community.
And so to that end, training core trainees, I would like you to each think about what data you are generating and what data you might need to organize, and come with an idea of that and some questions for Anthony for the next week's training. Okay, so that's your assignment, is to really think about the data, type of data you're generating, and what you're going to have to do to organize it. I really relate, Nirav, to the slide you showed on naming your files. I have learned through sad experience that if you don't name them properly, it's really hard to find them again, right?
Yes.
I have one more question, and then hopefully students will have thought of some questions as well. You mentioned the Sweeter project at the very beginning of your talk. And you mentioned that you're working with Berkeley and with New Mexico. And yes. I just wondered if you're working with the Superfund Center at Berkeley, or is it outside the Superfund?
It's their Data Science Institute. And Texas A&M is the primary on it, and then Texas Advanced Computing Center, who's like my partner in Cybers. Okay. So it's mainly the technical teams of it, but in terms of our outreach, we actually have funding for seed grants where we pair people up. And if there's something that we can intersect with the SRPs, that would be, we would be very open to it.
Berkeley has a center, Texas A&M has a center. And what university are you working with in New Mexico?
I think it's New Mexico State.
Okay, so I think it's University of New Mexico that has a Superfund Center there.
Okay. So we could very much think of, I know Satish graduates in May, but if we had to do a training, we could use Texas A&M and Berkeley and some of the Sweeter people there to be part of a training activity for the Superfund, then we could definitely think of a joint exercise.
Yeah, I think that would be really cool. I know MIT has led this last year in putting on a seminar program which I participated in, where they're having directors come in and talk about research at each of the centers. Very enlightening, really. And I think if we could do the same thing for the data science, to sort of create a larger group and not be so insular, it would really benefit the Superfund community.
Yeah, I think that's a great idea, Reena, because if we can get, the last time when I attended one of those sessions where they had breakout groups, there was a lot of interest in the fundamentals of these topics and training. And there's so much material we have in Cybers and others that it's not too hard to do that. And Michelle is a certified software carpentry instructor. So we have the ability to do some of these activities.
Yeah.
But it would be great if some of our own students could be partners in it. Then it makes it even more exciting for us.
Yes, I agree. All right, so I have hogged the questions. And students, do you have any questions? Trainees, do you have any questions?
Hi, thanks for your presentation. I had a question about the slide that showed the showing the fair practices for publications. The bullet point about including a section in papers about how to make things reproducible. And I was wondering if that was referring to like a written section in the methods, or like what an example of that would be.
I can definitely send you some examples, but yes, it would be in your methods where you say, the availability of this is here. And typically what people end up doing is they give you all the tools to make it reproducible, including the sample data, and then they tell you how to, the steps that are required to reproduce those. Since you asked this question, which I'm glad you did, the more modern and contemporary way of dealing with it is a lot of this analysis is put into something called continuous analysis and integration. And so these are programs that are constantly running, but they only run when a change happens. So when you, let's say, update your script to calculate something, your R program, when you put a final copy of it, it'll run your whole pipeline because there's a workflow engine behind it. And so anytime a change happens, it actually takes your sample program, it puts the data through it. And if the output doesn't match, it'll let the people know. And you can have any number of people watching it because it's open. And so if Reina wants to use what I created and I'm going to modify something, it might mess up Reina's analysis if she gets my latest version. So if this latest version works for Reina, then she would just get a notification saying, hey, Nirav made a change, but your analysis results didn't change. So it's good. Now it's important to have that kind of automation, but that's sort of still fairly complex for people, but that's what people are now building is the continuous analysis and integration where they watch the Git repository for change and that triggers the reanalysis. And so the machines are doing the reanalysis. It's not even humans. And they alert you when things are not, the results don't match. So you can go figure out where things are breaking.
Okay. Thank you. I was also wondering, so I have attended a couple of the software carpentry workshops, but if I
Very
wanted
good.
to revisit that material, is that all available on their website?
Yes.
Okay.
Yes. So if you go to software carpentry, so I don't know, when was the last time you attended one? Like how long ago?
I don't remember which month, but it was this past.
But like a year ago, maybe?
Yeah, around there.
A year ago. So if you can find the email that was like your confirmation or something, there'll be a website that they would have listed. And if you can't, don't worry about it. If you just go to like software carpentry, there are lesson plans. The only reason I asked you to look up that email is the instructors have a little bit more freedom in tweaking the content. So they may have added things that are relevant to your local institution. There might be things about your campus supercomputer or something in cybers that they'll ask you to use. So if you can find that, good enough. If not, just go to the software carpentry website and under lessons, they have them nicely cataloged. Pick up the lesson for what you need, and then all of the details are there. And if you have any trouble, just drop me an email and I'll make sure you're able to find it.
And that software carpentry would be in cybers?
Software carpentry is actually an organization to itself.
Separate, okay.
Yeah, so it was a movement that Greg Wilson started a long time ago in the 90s to help scientists who were writing software, but were not comfortable sharing it because they felt it wasn't good enough. And he sort of wanted to help them make it better. And there are thousands of workshops at any given month across the world in software carpentry. Cool. And it's all volunteers. Any other questions?
Great questions, Kendra.
Yeah, Kendra, those were good questions. Thank you.
Any other questions? Okay, so remember your assignment for next week. Think about the data sets that you have because we're gonna try and incorporate that into Anthony's presentation. Trying to understand what to do with your data sets.
Yeah, and Anthony, is our in-house expert on many things, including the statistics part of it. So he's going to give you a nice fly through on some techniques that are useful, and then you can chat with him on whether it's designing experiments or analyzing it, reproducibility. So please feel free to get in touch with us. We want you guys to be the most awesome papers that come out of SRP, and we want the journals to say, yes, this comes from this group and it is reproducible and we have verified it.
Yeah.
I want that badge of honor for all of you guys.
Thank you. That was well said, Nirav. And also, the DMAC is a core for our Superfund Center. And that means that it is available to all of you. And its resources are available to all of you. And I think Anthony will be your first, and Michelle, I guess, as well, will be the people that you first interface with. And so I think our next meeting,
maybe you can show them the place where they can go ask questions or contact you. So we've, as part of the DMAC core, created a place where people can go ask questions, so that way it's cataloged and we can all answer. So yes, we are a resource and we have put together some tools to make sure we can streamline the communication and help each other.
Yep. All right, Nirav, thank you so much. That was excellent. And Anthony, we look forward to seeing you next week. And with that, we'll adjourn.
Thank you.
Yeah.
Thank you. Bye, everyone. Have a good evening.
Thank you.
Bye.
That was good.